There's a new technology that is available for vision science, which is using stem cells from an actual patient. So from a clinical perspective, what we do is we can take a very small sample of skin from them, and we can convert their skin cells into stem cells, as you saw in the talk. And then from those stem cells, we can actually grow very early developing eyes. And from our previous studies, what we've seen is the PAX6 gene begins to work very, very, very early on in eye development. It's one of the very first genes that actually starts to tell cells to become the eye. It sends signals controlling other genes, and there's various cascades that then help the retina to differentiate into the multi-cell layer organ that it is. And so what we're planning to do is grow these stem cell derived eye cups from patients with aniridia and then their sort of normal healthy siblings or age match control individuals, so that we can compare what's happening with the aniridia eye to a normal healthy eye and using it as a model to test potential drug treatments. So essentially that's the kind of study we're going to be doing, is growing up those eye cups to use as models for understanding aniridia, but also to test new drug treatments and potentially in the future go on to maybe develop gene therapy. I've seen a couple of questions flash up and disappear. Maybe I could help answer those.
Yeah, okay. So Angela, who is in New York, asks, what about patients who have other types of mutations, not nonsense ones? Is it less likely there will be these sorts of medical treatments available?
So there's a lot of advances being made, not only for these nonsense mutations, but another type of mutation called a splice site mutation. There are forms of drugs that particularly act on that. A splice site mutation, they're quite complicated sort of mutations, but if you can imagine your gene is made up of subunits. Certain subunits give the instructions to make a protein, and in between those subunits are thought to be strings of genetic code that don't really make much sense. Now, when a gene is being made into a protein, all those strings of letters are cut out and just the instruction parts are put together. And so some patients have something called splice site mutations, which are present at the boundaries of these subunits. And so when your sort of cell machinery goes to cut out the junk genetic code, because there is a genetic mutation there, it doesn't quite do it properly. And that's why you end up with aniridia from that particular type of mutation. There are some now drug companies who are developing other forms of small molecule drugs that target splice site mutations that allow the sort of interfering genetic code that wasn't cut out to be cut out appropriately. So that may be developed for aniridia in the future. The other thing that I touched on just when I was sort of talking about the project we're about to start with Fight for Sight and Aniridia Network UK is potentially a gene therapy for PAX6. Gene therapies, as I've mentioned in the talk, are becoming a reality for patients with inherited retinal diseases. The technology is there, the viral vectors, non-viral vectors for delivering genes. So I think one of the things we should be looking to the future for is actually developing a gene therapy for PAX6, which means that any patient with whatever type of mutation would benefit from this treatment. And obviously, it's questionable about when do you deliver this treatment. Most patients will obviously receive this after birth. So you may not need to deliver it into the eye as such. If we could develop some form of gene therapy which could be delivered as an eye drop so that it reached and penetrated the cornea and potentially into the front chamber of the eye, then we could help to prevent the cornea becoming cloudy in the future. We may prevent the risk of cataracts and glaucoma developing. So that's something to look forward to. Another thing I touched upon in the talk again was high throughput drug screening. So We have hundreds and thousands of drugs that we use daily for treating various different conditions. Those drugs are all approved. They've gone through safety trials. We know what their side effects are. We know how they work. But what we're finding now is that some of those genes had functions that we didn't know that they had. And so quite a popular form of finding treatments is to screen these known drugs to see if any of them have any potential benefit for aniridia. So if we take the example of those zebrafish that I showed, you have an aniridia zebrafish, which clearly shows there's a difference in the formation of its eyes compared to a normal zebrafish. If you tested all the drugs on the aniridia zebrafish, and then you saw with maybe 2 of those drugs that actually the eye looked much healthier, the structure of it looked more normal, you could go on to investigate that particular drug further. You could test it on those stem cell eye models. And then if it was shown to be successful, we would be able to what we call repurpose that drug and apply it for aniridia as a treatment in a clinical trial to see if it had any benefit. And for that, it would potentially work on any patient with aniridia rather than focusing on a particular type of mutation. I hope that answered your question.
Marvelous. And then the other question here was, do I understand correctly that if approved, these drugs will only slow down things getting worse in adults? They won't boost sight, but in children, things might improve. Also, will it affect all parts of the eye, or will it help certain parts of the eye, such as cornea, etc.?
Okay, so if we're going to talk about atalurin and potentially amlexanox, both of these drugs, particularly atalurin, is given as a powder which you dissolve in water or some sort of solution, and you drink the drug. So you're taking it orally, which means that that drug is essentially passing through your bloodstream and reaching every part of your body that the drug can get to. We know from some of the work that we've done with other disease models and with the company that the drug atalurin can actually enter into the eye in high enough levels to have a beneficial effect. So if a drug is given orally and it reaches the eye, the back of the eye, the front of the eye, then it could have an effect on the whole eye. If, for example, a drug was just delivered to a particular area of the eye, for example, conventional viral gene therapy, patients receive an injection underneath their retina, or if the drug was packaged to be an eye drop, then yes, you could say that the drug may just target a specific tissue like the cornea or the retina. So that answers a sort of delivery. That's more of a delivery mechanism. With regards to would it help to slow degeneration down or reverse it? So obviously in childhood, the eye has less complications of the aniridia. So when the child is born, they do lack the iris to a variable degree. The back of their eye hasn't completely remodeled. So that remodeling process is still underway for the first year of life. So there is a potential that if you gave the drug early enough, it would help with that developmental process and may help to improve the vision. But if you have a clear cornea and you don't have any cataracts there or glaucoma, then if you're giving that drug very early on and it's producing PAC6 in enough levels to keep the cells healthy, then you could prevent further disease or you could just slow the process down. So when it comes to adults, if you have somebody that already has established corneal scarring or glaucoma, it's harder to reverse that because where you have scar tissue or dead cells, you cannot, in some tissues, regenerate that. You cannot, no matter how much protein you produce, if the cell is just not functioning, it's not working, or it's scarred, it's not going to revive itself. So these treatments are very much for where you have cells that are present that may not be healthy, but are still able to function or survive, you can deliver that treatment, it can produce the PAX6 protein, and it will help it work better. So really, those drugs are for an earlier stage where you haven't got scarring and the destruction of cells. Where you are in that situation, you may need to consider other forms of treatment, whether that be limbal stem cells, other kinds of treatment that improve the window to the eye. But potentially, if, for example, you had a graft or something else, if you were to give the drug alongside that, then any remaining cells that you have, you could help to produce the PAX6 protein that may help protect and reduce any side effects from the treatment.
Last question here was, would these drugs potentially help other PAX6 related conditions other than Aniridia?
So other conditions caused by PAX6? The answer is yes, depending what those were.
So I'm guessing things like Peter's anomaly or anything like that, sort of other sort of related.
Yeah. So it depends on the condition and it depends on the mutation. So as I mentioned, the great thing about the drugs are, well, at least this class of drugs, is that it works on the mutation and it doesn't really matter what condition you have. However, if the condition is a structural eye defect, so we know that, for example, PAX6 can also cause microphthalmia. It could be related to, as you said, Peter's anomaly, etc. If you have structural defects, it's not going to change the structure because we're talking about a postnatal treatment. You would have to be giving these drugs very early on in pregnancy, and we haven't broached that sort of area yet for it to have an effect. Because that's the time, in the first 2 months of pregnancy, is when the eye is developing appropriately. So a treatment would either need to be given very early on at that stage, but potentially complications from those structural defects later on in life could be reduced by these types of drugs.
Great stuff. We need to move on. So I'd like to, on behalf of everyone here, thank Mariya hugely for her time, for coming and answering questions today and getting sort of respect for her speech at Conference 2018. Yeah, thank you very much.
Thank you.
All right. Cheers. Take care of yourself. Goodbye.
Bye.